The overall goal of this procedure is to evoke and analyze sodium dynamics in small cellular compartments such as spines and dendrites of central neurons in the intact tissue. This is accomplished by first preparing an acute mouse hippocampal slice, loading a pyramidal neuron with the sodium-sensitive fluorescent dye SBFI through a patch pipette, and visualizing the cellular morphology using multi-photon excitation. The next steps are to choose a spiny dendrite for the experiment, and to focally inject a photoactivated compound, such as caged glutamate, close to this dendrite. A UV flash is then applied to focally uncage glutamate using a UV laser that has been accurately positioned to target a small volume close to the dendrite of choice. The final step is to monitor the resulting changes in SBFI fluorescence in the dendrite and to monitor the resulting changes in sodium in the adjacent spines. The accompanying membrane currents are also recorded. Ultimately, using the appropriate pharmacological tools such as receptor blockers will enable the study of the mechanisms generating the evoked intracellular sodium signals.
This method provides a reliable tool for the investigation of intracellular sodium signals in small cellular compartments and in intact brain tissue.
Here, we combine whole cell patch clamp and multi-photon sodium imaging with a modified procedure for UV light-induced uncaging. This allows us to monitor cellular responses upon precise and focal applications of neuro-active compounds.
The procedure will be demonstrated by Karl Kafitz and by Kleinhans, a grad student in my lab.